Okay, now we're going to talk about when that genetic code changes or a mutation happens. And this is the driving force of evolution. And what that means is that the nucleotide sequence changes. There's the wild type where the microorganism has this natural non-mutated characteristic. And then, like you have up here at the top, and then you have the mutant strain where the organism starts to show some sort of mutation. Like here, it's continuing to produce offspring that resist antibiotics to treat it. Here, if given the antibiotic, it kills it. But here, for some reason, they're not making the enzyme that's needed for the antibiotic to work. Spontaneous mutation is just a random change in their DNA because of errors in replication. And induced mutation is because they are exposed to mutagens, which are usually chemicals or radiation. These are the categories of mutations. Point mutations is where bases are added or deleted or substituted, just a few of them. A missense mutation causes a change in one amino acid only, and a nonsense mutation changes a normal codon into a stop codon. A silent mutation alters a base but doesn't change the amino acid. A back mutation is when a mutated gene reverses to its original base composition. And a frame shift mutation is when the reading frame of the messenger RNA is altered. Now, we're going to go through the effects of the 5 major types of mutations. First, there's the wild type, which is the non-mutated sequence. For instance, the big bad cat ate the fat red bug. The wild type sequence of this DNA sequence that's found in organisms is just considered the normal sequence. Okay? The big bad cat ate the fat red bug. That's the normal non-mutated sequence. But if we have a substitution mutation like missense, the big mad cat ate the fat red bug. Instead of the big bad cat, it's the big mad cat. You have a different amino acid incorporated into the protein, and the effects can be severe. It's based on, or maybe unnoticeable, just how much it alters the protein function. The third type is a nonsense, the big bad XXX stop. It converts a codon to a stop codon. So we've not completed making our protein, and usually this is going to be a severe mutation. Inversion, the big ABD cat, because we inverted B and A, ate the fat red bug, or the bib gad, we inverted the G and the B. Cat ate the fat red bug. This is when the adjacent letters change places, and that can alter 1 or 2 bases depending on where it happens. In the first line up there of the example, it happened in 1 location. In the second, it happened in 2. That doesn't change the reading frame, but it causes changes in the amino acids that are made and the proteins. Now, a frame shift mutation, you can have insertion, the big BAB, and then you've got all these others inserted, or it can be deleted, the big, okay? So insertion adds a letter, deletion removes it. That causes the whole reading frame to be changed. Because you see what happened in the first example, insertion, the big ba, you see how the B was exchanged for D? And even though the rest of it, if you break it up, it says cat ate the big red bug, it's changed the whole frame. So it results in a different protein where every amino acid after that mutation is affected. So this is usually going to produce a non-functional protein. Mutations can be fatal often, so the cell has some enzymatic repair mechanisms to find these mutations and try to repair the damaged DNA. DNA polymerase proofreads the nucleotides during DNA replication. It also helps to locate and repair mismatched nitrogen bases. Light repair happens for UV light damage, and then there's excision repair, where a series of enzymes locate and repair these incorrect sequences by removing them and adding them where they need to be put in the correct nucleotides.